Cellular respiration is a process that breaks down macromolecules to produce ATP. It can be done with lipids, proteins, but more often than not, it's done with carbohydrates, specifically glucose. So that's what we're going to focus on in this video. It's currently July 8th, Corona Summer 2020, and let's get started with this. Okay, there's three processes to cellular respiration. One is glycolysis. The second is the Krebs cycle, otherwise known as the citric acid cycle. And the third involves the electron transport chain and chemiosmosis. You could include oxidative phosphorylation within that description, but we're just going to suggest electron transport chain and chemiosmosis. This video is going to focus on glycolysis and the Krebs cycle, and then I'll have a subsequent one specifically for the electron transport chain. Ultimately, the goal of cellular respiration is to break down biomolecules, in this case glucose, into a stepwise fashion, harnessing as much energy as possible and transferring it to the ATP molecule. ATP is what we're going to use for energy throughout our body to do work. We could break down glucose in one big step, but we're going to lose a bunch of energy that we can't use. So let's see how we go about this. So the first process is glycolysis, using the 6-carbon molecule of glucose. At the end of glycolysis, we end up with 2 molecules of pyruvate. Pyruvate are 3-carbon molecules. So if we break down glucose, just essentially divide it in half, we're going to have 2 3-carbon molecules known as pyruvate. At the end of glycolysis, we also have produced 2 ATP molecules, and I should suggest it's 2 net ATP because glycolysis produces 4 ATP molecules, but it utilizes 2 of those ATP molecules in the process. Glycolysis is a 10 enzymatic step process, and a few of those steps actually use ATP. So in the end of glycolysis, we're left with just 2 ATP molecules, and we are also left with 2 NADH molecules. Nicotinamide adenine dinucleotide is NAD, and we've added 2 hydrogens to it. So a couple things to talk about here. One, NADH and another molecule that we'll talk about in the next slide, FADH2, are shuttling electrons from glycolysis in the Krebs cycle to the electron transport chain. And that process, or the movement of electrons by means of NADH and FADH2, is arguably the single most important aspect to glycolysis and the Krebs cycle. The goal here is to make ATP, but we're only making 2 ATP via glycolysis, and as we'll see in the next slide, only 2 in the Krebs cycle. But we're making a ton of ATP via the electron transport chain. And that can't be done without the use of NADH and FADH2, which are functioning as shuttles for the electrons to the electron transport chain. And with that said, really the whole purpose of this whole process is to liberate electrons from the hydrogen atoms from this biomolecule, in this case glucose, to utilize those electrons to activate the electron transport chain that once again we'll talk about in detail. So at the end of glycolysis, we have 2 pyruvate molecules, 2 ATP molecules, and 2 NADH molecules. So what happens after glycolysis? Well, that really depends on one thing, and that is the presence or absence of oxygen. If oxygen is present, that's going to determine the fate of pyruvate. If oxygen is present, pyruvate is going to be converted to acetyl coenzyme A, and we talked about that molecule earlier when we were talking about acetylcholine. So pyruvate, in the presence of oxygen, produces acetyl coenzyme A. The conversion of pyruvate to acetyl coenzyme A creates carbon dioxide. So in this slide, as you can see, I have 2 acetyl coenzyme A and 2 carbon dioxide, and that's because we will have 2 pyruvate molecules at the end of glycolysis. So the conversion of those two pyruvate molecules and acetyl coenzyme A results in 2 and 2 right here. And pyruvate needs to be converted into acetyl coenzyme A so it can enter the Krebs cycle. To be clear, pyruvate, which is the end product of glycolysis, does not itself enter the Krebs cycle. It needs to be converted to acetyl coenzyme A. In the absence of oxygen, pyruvate enters a process of fermentation, specifically lactic acid fermentation, which we're not going to talk about in this video. The citric acid cycle creates 2 carbon dioxide, 1 ATP, 3 NADH, and 1 FADH2. Once again, NADH and FADH2 are going to shuttle electrons to the electron transport chain. But a cycle happens once. It's one cycle through. We have 2 acetyl-CoA molecules going through this cycle. So we have to multiply all of this on the left by 2. So we're going to end up at the end of the citric acid cycle 4 carbon dioxide, 2 ATP, 6 NADH molecules, and 2 FADH2 molecules. Keep in mind, I've just said there's 4 CO2 molecules created in the citric acid cycle, and just previously I suggested there were 2 CO2 molecules created in the conversion of pyruvate to acetyl coenzyme A. That is 6 CO2 molecules, and that's where you get the 6 CO2 in that cellular respiration equation that we've memorized. The CO2 for our purposes is not important. I've been saying all along it's just a byproduct. The goal is the production of ATP. Left with 4 ATP molecules, 8 NADH molecules, 2 FADH2 molecules, and that's it. And 4 ATP is really not that much compared to how many ATP we are going to produce in the electron transport chain. So the important molecules here are really 8 NADH and 2 FADH2, which ultimately are going to be converted to about 34 ATP molecules in the electron transport chain. One thing I just want to clarify, the electron transport chain in chemiosmosis occurs in the mitochondria, specifically in a region known as the inner mitochondrial membrane. The Krebs cycle occurs in the mitochondria, specifically in an area known as the matrix, the mitochondrial matrix. Glycolysis happens in the cytosol of the cell, not in the mitochondria. As a result, there are some cells in our body, for example, red blood cells, that do not have mitochondria. And the only way they can make ATP is via glycolysis. It doesn't matter if oxygen is present. Generally, it's not. They make ATP anaerobically in glycolysis. So what happens after that? The electron transport chain in chemiosmosis. I'm going to save that for an entire lecture unto itself, and we'll talk about that shortly.